Welcome everyone, and thank you for joining us. I'm Brenda Dettinger from BioPAC, and I'll be your moderator on today's webinar, Advancing Rodent Electrophysiology with the EPOCH Wireless Telemetry System. Today's webinar is being recorded, and we'll send each of you a link once the recording is processed. During the webinar, if you have questions, please submit them through the GoToWebinar control panel. That's how you communicate with me, your moderator, and I'll pass as many questions on as I can to our panelists. Now, on to our presenters. I have two fabulous experts today. I have Dr. Andrew Zayachkivsky, and he is a neuroscientist and assistant professor currently working at the University of Utah. He earned his BS from Allegheny College, his neuroscience PhD from the University of Utah, and he completed his postdoctoral fellowship at Yale University. Andrew helped develop the EPOCH system during his PhD work and can answer any questions related to this technology. We also have Fraser Finlay, the CEO of BioPAC. He has over 25 years of experience in the life sciences and physiology monitoring industry. He's a well-regarded expert who presents at workshops all over the world. Fraser, let's start with you. I'm making you presenter.
Great, thanks Brenda for the introduction. Welcome everyone to this EPOCH Small Animal Telemetry webinar. A little background information about BioPAC. BioPAC Systems is a global leader in life science research solutions, empowering scientists to measure physiology anytime, anywhere, and from any subject. BioPAC has been advancing research for over 30 years with its cutting-edge tools and software trusted by 99 of the top 100 universities worldwide. With more than 57,000 scientific citations, BioPAC's technology is also relied upon by leading tech companies for product development, validation, and human-centered design. So today's webinar is all about the EPOCH Small Animal Telemetry System. This is a relatively straightforward system. It's a skull-mounted transmitter that wirelessly telemeters the signal to a receiver tray. One of the unique features of the system is the actual size, and the system was developed at the University of Utah by researchers, Andrew being one of those. They needed a system to advance their research and understanding of neonatal and childhood seizures. BioPAC has licensed the intellectual property from the University of Utah and manufactures the system. At the heart of the system and why the system is suitable for small animals, particularly rodent pups, is an ASIC. An ASIC is an application-specific integrated circuit that is unique to this particular device. It's remarkably small and very power efficient, which makes it ideal for the small transmitters that are needed to operate continuously for long periods of time. Most commercial systems that use telemetry, wireless telemetry, will be developed utilizing off-the-shelf components. The advantage of that is they're usually relatively inexpensive, they're readily available, and that works well for low volume production runs. But the downside to that is they're usually quite a bit larger than you would ideally like, and they're not as power efficient as certainly the ASIC that is part of the EPOCH system. And that ASIC, with it being so small, it allows us to offer the smallest wireless transmitter suitable for, mice pups, so really small. The system is actually quite straightforward. You have a receiver tray, which is shown on the left-hand side over here. the transmitter, which is mounted on the skull of the animal. And the system comes with an activator, and Andrew will demonstrate this later on, but that's essentially it. We use a capacitive coupled transmission scheme. The system will accommodate 2, 4, or 6 channels of data from the animal back to the receiver tray. So I've already mentioned one of the significant advantages of the EPOCH system is the ability to record data from rodent pups, particularly mice as young as P10 and rats at P6. This is critical for anyone wanting to study childhood diseases using age-appropriate models. Now, this is the total layout. On the right-hand side over here, you have a computer that's going to collect all of your data, monitor to display what's going on. An HD system, so here, this is the MP200 system, 16 channels. This is the MP36, which is 4 channels. And we've got the receiver tray, the activator, the transmitter, and then on the back side of the receiver tray, which is not shown in this picture here, you have a bank of BNC connectors, one BNC connector per channel of data. This provides an analog output, which with the appropriate connector will interface between the receiver tray and the data acquisition system. So it's a relatively straightforward setup. You can run multiple receiver trays back to one MP200 system, but in addition to that, you can run multiple MP200s to one computer. So this makes it really easy for workflow efficiency, specifically for technicians and experimenters that are looking after a lot of setups. The trays will work nicely with commercial racking systems, and here if you look carefully you can see the Faraday cages just around these. Each system comes with a Faraday cage. Now some of you will be fortunate enough where you don't need to use them, but if you are in an environment that's a little bit noisy and you've got a lot of stray RF or EMF signals, this will help shield those. So the bottom is the receiver tray, this is the animal cage, and then the Faraday cage that comes with the system. In terms of the transmitters, there are 2 basic types, single-use and reusable. What we're looking at here is one of the single-use devices. There are 3 sizes. One that is used for the pups, which will last for 2 weeks of continuous recording, and this is used for EEG only. And there's 2 larger ones, one for adult mice and one for adult rats. Now, you can use the adult mouse one on the adult rats if you want. The adult mouse unit will provide you with about 2 months of continuous use. The larger rat version will provide you with 6 months of continuous use. 2 channel for the pups, 2, 4, and 6 for the adults. When you receive the transmitters, they come on a little circuit board. They're attached to it. This enables us to ship them disabled. Using the activator, you plug the board in with the transmitter mounted on it, and you turn the transmitter on and you can activate it into a test mode whereby you place the activator on top of a receiver tray and it will output a constant signal. And that just allows you to confirm that everything is working before you perform the surgery. And Andrew will walk through this in his segment. The second type of transmitter is a reusable one. This is super helpful because it allows you to turn the transmitter on and off so you can increase the battery life. The battery life is actually 2 months, but that's 2 months of continuous use, and you can obviously run it longer than that. You can also share it across animals. So if you've got 3 animals implanted, you can use one transmitter, and although you can't record simultaneously, you can use that one transmitter to record data from all 3 animals. The other reason why this is super helpful, if you need to go beyond the typical battery duration, you can replace the transmitter on the animal. So you just unscrew it, drop a new one on, and you're basically continuing to record beyond the life of a single battery. This image here is actually showing one of the Plastics One electrodes. Plastics One used to make a really nice little pedestal that enabled us to connect to this really easily. They no longer offer that. So we've redesigned this, and it's allowed us to actually reduce the profile of the entire unit so it doesn't stand quite as proud as it used to, which makes it a little bit easier, better for the animals in their cage. But again, 2 types, single use and reusable, and this is the reusable option. In terms of the signals that can be recorded, EEG and ECG, EEG EMG, ECG EMG, and 2 channels of EMG. Specifications, the green relates to the 2 channel. The important things to note here, if you can use this with a common reference or differentially. So if you're using a differential setup, you will use 4 electrodes. If you're using a common reference, it will only be 3 electrodes. For the 4 or 6 channel option, only available as a common reference. Here you'll have 5 or 7 electrodes. And then the input impedance is 5 megaohms. The noise is pretty good, less than 7 microvolts RMS. The units ship with a standard 2000 gain, but at the point of ordering, you can select a higher gain if you're using it with pups, or you can reduce the gain for different applications. This is the footprint of the electrodes. So this is the 2-month and 6-month dual channel. So if you're using a common reference, channel 1 and 2 back to the common reference. If you're doing differential, you've got channel 1 back to the channel 1 reference, channel 2 back to the channel 2 reference. For the pups, you've basically got 2 channels of EEG back to the common reference. For the 4 and 6 channels, only available with a common reference, so the same reference whether it's 4 or 6. Here showing the 4 electrodes, here showing the 6. Okay, with that said, I'm going to hand over to Brenda before Andrew walks you through the surgical part of the system. So Brenda, over to you.
All right, thanks so much, Fraser. Andrew, go ahead and make you presenter.
Thank you, Brenda. So in this presentation, I'd like to show everybody the surgical procedure for implanting a transmitter into an adult rat. Having worked with and trained other researchers in various environments, I found that surgical mistakes are probably the #1 source of failures of transmitters or recordings. So in this video, I wanted to go through the surgical procedures to ensure good practices during surgeries, so that all of us who do recordings with these animals get beautiful traces and good recordings. Here I have the video of the surgical procedure. This is an adult rat implanted with an EPOCH transmitter. The first step in the procedure is to activate the transmitter. To activate the transmitter, insert the transmitter that's still on the activation port into the activator, and power on the activator. The activator has two buttons, power and activate. Power button turns on the activator. You see a light up in here. And activate button activates the transmitter. Sometimes you have to hold activate button for several seconds for the transmitter to activate. Next step, we want to test whether the transmitter is communicating with the receiver. So to do that, we place the activator and the transmitter onto the receiver and wait for the signal light to turn on. Remove the transmitter from the receiver to make sure the light turns off when you remove it. This is a typical surgical setup using gas anesthesia. This is the way my colleagues do it in the lab. Your setup may be different. Please follow your guidelines of your IACUC for your surgeries and surgical procedures. So for tools, we will use spatula, forceps, scalpel, scissors, hemostats. For consumables, we will use iLube, glue, cyanoacrylate glue, cauterizer, and cyanoacrylate glue accelerator. Use your institution's guidelines to sterilize the transmitters. Here we sterilize transmitters using chemical sterilization. So the first step in the surgery is to prepare the animal. In order to prepare the animal has to be anesthetized. Place the animal in the induction chamber, set your anesthesia levels to what's described in your protocols, and make sure oxygen is flowing at a specified rate. After the animal is induced, remove the animal from the induction chamber. Place the animal in the nose cone with anesthesia still flowing to maintain the appropriate levels of anesthesia, and apply iLube to the eyes of the animal. This is particularly important in dry environments because animals' eyes dry out pretty quickly when anesthetized. After applying iLube, shave the head or remove the hair to make a clean incision site that we'll use for the surgical procedure. It's important to remove the hair because for closure of the wound later on. After the hair is removed from the animal's head, place the animal into the stereotactic frame by inserting the ear bars into the ear canal and tightening them at the appropriate level. After the animal is placed in the ear bars, the surgical area should be sterilized. Follow the guidelines of your institution to what's in your protocol. After the area is sterilized, we're ready to make an incision. Incision can be both small or large depending on the surgeon. I prefer to make my incisions larger than the surgeon in this video to enable good access to the skull. That helps ensure the better adhesion of the transmitter later on. After incision is made, use cotton swabs or gauze to soak up all the blood and scrape the periosteum from the surface of the skull. Following this, use hemostats to spread the skin on the surface of the skull to enable access to the surface where the transmitters will be mounted. Some surgeons prefer to use 4 hemostats, some surgeons prefer to use 2. Both methods are fine. In this case, 4 are being used. After the hemostats are placed, once again, wipe the surface of the skull and ensure that all the periosteum is removed. Oftentimes there is surface bleeding on the surface of the skull, so use cauterizer to control the bleeding in these localized areas. If there's a larger bleed, feel free to use the cauterizer to stop the bleeding as well. Here's another example of a surgeon scraping periosteum from the skull. So the next step is critical in the process. Here we must be careful not to cut electrodes too short because once we cut the part of the electrode off, it's impossible to put it back on. So it's better to err on the side of leaving more wire on the transmitter here and aligning before permanently cutting the wire to the length you'd like to use in your procedure. So in this case, the surgeon leaves about 8 millimeters of wire on the transmitter. Next, use the transmitter to mark the surface of the skull where the wire comes out. This is where we'll be drilling boreholes. You can use a cauterizer like surgeon here, or you can use a Sharpie. Make sure these holes are aligned because it makes it a lot easier later on to put on the wires in. Here, after the marks are made on the surface of the skull, burrs are used to drill holes into the surface, into the skull. It is important not to let the drill bit slip in and hit the surface of the brain. To do that, make sure that the surgeon's hands are very well anchored on the frame. Additionally, I prefer to use saline to lubricate the surface, which makes cutting quicker and does not heat up the skull as much. Again, do this step really cautiously and make sure that the drill bit doesn't penetrate the brain. After the skull electrode holes are drilled, place several screws, skull screws, on the periphery of the transmitter. These screws are particularly important for chronic implants where recordings are being made for 2 to 6 months, as they stabilize the transmitter on the skull long term. Use appropriate burr to drill the pilot hole for the screws. I prefer to use self-tapping screws in this case. Then screw the screws into the skull, again making sure it doesn't penetrate into the brain. After the skull screws are in, once again clean the surface of the skull from blood, and now the transmitter is ready to implant. Another step that's not shown in this video that some surgeons like to use is to use hydrogen peroxide to clean off and debride the bone, which makes the adhesion surface even cleaner. Next step is to start gradually cutting the electrodes like the surgeon is doing here and aligning them with the existing holes in the skull. Again, be cautious not to cut off the wire too much because once the wire is cut off, it's impossible to put back on. Leave about 1 to 2 millimeters at this point. You may need to cut it even shorter to make sure that there's no penetration of the dura. After the wires are cut, test fit the transmitter to the skull and make sure all the wires are going into the holes, like the surgeon is doing right here. The transmitter should be flush with the skull if all the wires go in. So one important step that the surgeon did not do in this video is to apply a bead of glue on the bottom of the transmitter. So I usually like to go around on the perimeter of the transmitter and put one line of glue in between the electrodes. Be very cautious when you do this because if the electrodes are contaminated with the glue, they will be insulated and no biopotentials could be recorded with insulated electrodes. After the transmitter is applied to the skull, put a bead of glue around it, like the surgeon is doing right here. Be very careful not to knock the transmitter out of its position in this case, especially if you have glue on the bottom of the transmitter. Once the bead is applied, make sure you also apply it around the screws as that anchors the whole implant better to the skull. After the bead is applied, use Loctite super glue accelerator or center acrylic accelerator to instantly harden the glue that's around the transmitter. Don't use too much of it as this is an irritant. After the accelerator is applied, wash the area with saline. Next step is to build a head cap with dental acrylic. This ensures very smooth surface for wound closure. And if the surface is smoother, the animal is much less likely to scratch at the transmitter and open up the wound later on. And this also offers additional adhesion, additional surface area, adhering the implant to the skull. In this step, make sure that the skull screws are completely covered because dental acrylic doesn't bind well to the bone. However, it binds really well to metal skull screws that are inserted into the skull. Wait several minutes for the acrylic to start hardening, and then make sure that the edges of the implant are smoothly attached to the skull. We don't want to create any extra ridges that will cause irritations later on. Next, make sure that the skin is retracted over the implant. We'll be suturing on the both front and the back of the implant to make sure that the closure is flush with the transmitter and that the head cap implant is under the skin. You can use both any style of suture that you're trained in. In this case, the surgeon is using interrupted suture, which I prefer to use as well. You can start in the back of the transmitter or in the front of the transmitter, depending on your surgical preference. Again, just do the standard suture like the surgeon is showing here. It's good to have the head cap covered by the skin, because what can happen later on is that if the head cap, the acrylic head cap is exposed, it often skin keeps getting retracted, and that opens up an area for inflammation and infection getting in later. And just to make sure that these implants last a long time, like in our application for epilepsy, we like to make the skin flush and tight with the transmitter. Be cautious, don't go too tight, as that can cause other problems. Here's an animal. After the surgery is done, place an animal into the recovery cage until it's ambulatory, and once the animal is ambulatory, it's ready to start the recordings, EEG recordings. In this case, we have an animal recovered and being ready to be placed into the recording chamber. You may start your EEG recordings immediately after the surgery once the animal is recovered. Here's what the EEG traces look like on the screen with an enabled transmitter and transmitter functioning properly. So in this next part of the presentation, I'd like to talk about my experience with EPOCH system as a user and as somebody that helped develop this system. So first of all, the system is, like you saw in the video, is extremely easy to implant surgically. The surgical procedures take about 15 minutes for an experienced user, and they are really rather easy to execute. With this system, we're able to implant rat pups as young as P5 and mice pups as young as P9. And really, what's limiting the age of the implantation is the skill of the surgeon and your institution's rules. We are not limited by animal weight for implantations like the other systems that are implanted internally inside of the animals in the IP cavity. This is the head stage style implant, so the animals that are implanted with this device, particular pups, are free to move around the cage and recover very quickly. The transmitters can be removable or reusable if you purchase the reusable system. Finally, there's no tether, which is extremely important when it comes to EEG recordings, because having no tether really reduces movement artifacts, which are caused by the wire moving around, and wire making the really soft skull in the pups flex around. Finally, we have a chamber designed for work with the pups, which will help control temperature, particularly in the pups which are too immature to thermoregulate. So here let's talk about several applications where EPOCH system was used. Like I said, the system was developed during my PhD work. Um, in this case, we were studying an animal model of neonatal encephalopathy and neonatal seizures. This really became a study-enabling device for these studies. At the time, very few recordings of neonatal seizures existed in the literature, and that was probably primarily due to technical limitation of recording EEG from really young, small animals. As many others, I first started with the wire, and I've discovered that this was not the most optimal way to record EEG. Like I said before, what happened was that the skull often flexed around and caused artifacts, particularly during the convulsion. So we had to design EPOCH to fix these problems and record good traces of neonatal seizures. So hypoxic-ischemic encephalopathy is a really devastating condition in human neonates. And here we have an animal model that models this disorder. So in order to induce hypoxic-ischemia, we use a technique developed by Rice-Vanucci. And in this case, the animal's carotid is ligated, and animal is placed for 1 to 2 hours into a chamber with low oxygen. This makes animal have seizures, and our main goal was to record these neonatal seizures during the administration of hypoxia. As you can see here, we have 2 traces with seizures that occur during administration of hypoxic mixture. You can see the seizures consist of spiking activity. Here we have 2 different examples, one with 1 dominant frequency and second with the 2 dominant frequencies. The traces are free of movement artifacts, and we're able to go in and quantify power from the recordings. Next, we wanted to look at background EEG because we wanted to identify some potential biomarkers to be able to detect some of these brain abnormalities early on. So as a first step, we went in and recorded from control red pops to see what are the EEG patterns that occur in these animals normally. And here we have the 2 traces, one recorded from postnatal day 7 animal, and the second one recorded from postnatal day 11 animal. At postnatal day 7, we see typical patterns described in that are also described in human neonates. The pattern is very discontinuous, with a burst of activities happening with quiet periods around these bursts. As the animal matures, the higher frequency activity comes online, and the trace becomes much more continuous, with much less discontinuity as seen at the earliest ages. Next, what we did, we wanted to go in and compare the patterns from control animals with animals which had this hypoxic-ischemic encephalopathy. And what we saw that in the controls, the pattern that described in this previous slide was present, where the recordings becomes much more continuous with age, and higher frequency activity comes in line. However, animals hypoxia-ischemia did not have this pattern present. Instead, what we saw was rather almost isoelectric activity early on, and then the normal development of the normal EEG patterns never really occurred, with EEG patterns stayed very discontinuous, and higher frequency activity never come online. We're able to then use power spectral density plots to quantify this. So in this case, we have a figure where we looked at the ages that are on the left, and we quantify activity from control and animals with hypoxic-ischemic encephalopathy. And we see that here in black, we have animals with hypoxic-ischemic encephalopathy, and their patterns are never developed to the levels of normal animals. So this technique can really be used as a quantitative EEG biomarker for negative outcome, which can be rapidly detected early on after the infarct. Next application was performed by Chris Dula lab at Tufts, where they implanted the young mice with a 2-week transmitter to study infantile spasm, another devastating disorder seen in young humans and children. So infantile spasm consists of stereotyped sets of behaviors, which include flexion and extension and back arcing, and a very specific EEG discharge. So using our transmitter, Chris Dula and his lab were able to detect these changes and quantify these changes. Next application is for recording and monitoring of chronic epilepsy. So while the previous applications looked at more acute patterns, this looks more at chronic monitoring and chronic patterns seen in these animals. So this study was done by Jace Pompanato and Ed Dudek. And what they did here was implant the animals, monitor them 2 weeks, and then ablate interneurons in the hippocampus, and then look whether these animals developed epilepsy afterwards. And it looks like they were able to, record clean baselines, and then subsequently monitor animals for up to 74 days, and they were able to detect seizures which occur after interneuron ablation. Next application that the system was used for was to study status epilepticus in immature models of adults. Status epilepticus is a seizure that lasts for tens of minutes, up to several hours. It's a really kind of devastating condition that can cause death. So it's really important to be able to identify various pharmaceuticals that can stop status epilepticus without harming the animal. So what Erica Scholl worked on is to screen some of the compounds which were to stop status epilepticus. And here in the traces we see compressed waveforms where seizures occur for several hours. And once again, the recordings are relatively movement artifacts free and clean and have very good signal to noise ratio. Next, our transmitters can be used for monitoring cardiogram and myogram, so ECG and EMG. There are configurations available of EEG and ECG and ECG and EMG, or EEG and EMG. In this case, we see some traces which were recorded from rats with electrodes placed over the chest cavity. So these are the ECG traces. As you can see, you can see beautiful recordings. The next application is EMG, so the myogram of the animals. Here we have a combination of EEG and EMG on the same transmitter, and we see muscle activity, and there's no movement artifact on the EEG channels, showing that while the animal is moving, the EEG still remains clean.
All right, Andrew. So there was a bunch of questions that came in your section. So I'm just going to take a few minutes to go ahead and ask them now while your content is still pretty fresh. So tell us, what is the electrode material?
So for EEG, we use platinum iridium wires, which are insulated or not insulated. For ECG and EMG, we use a braided wire, which is made out of stainless steel. So the braided wire makes it easy to implant into the muscle because it's very flexible, and platinum iridium is biocompatible with the brain.
Okay, great. Thank you. Do you need bone screws when mounting the transmitter?
So bone screws are optional. Bone screws are good to use during chronic applications. For example, if you're trying to record epileptic seizures and the animal has to be implanted for weeks to months, it's good to have these skull screws because they really stabilize the transmitter to the brain. However, if you're recording from the pups, oftentimes there's simply not enough room to put the screws in and the skull is too soft. So in that case, you can just use cyanoacrylate glue without screws, but make sure that the skull surface is really clean to ensure good adhesion.
Okay, thank you. And then you showed examples of shorter monitoring. Can you also run longer studies, like 6 months or more?
Yes, we can certainly run longer studies, 6 months or more. What I would use then is the reusable transmitter, where the pedestal for the transmitter can be implanted in the animal's skull, and the user can then replace the transmitter as the battery runs out.
Okay. And is there flexibility in the location of these electrodes, or are they largely fixed in place?
So electrodes are fixed in place on the one-time use transmitters. However, the user can bend them and shape them into the location where they'd like to. You can make an L shape underneath the transmitter and then run it down and make up a place where the transmitter will be implanted. With the reusable transmitter, we also offer an option to for the user to implant just the wire and then run the wire into the implant and attach it to the uh, pedestal. So in that case, the user can really target their electrodes precisely in the specific brain structures where they would like them to be implanted.
Okay, are the coordinates adjustable or standardized based on the transmitter?
The coordinates are adjustable. So like I said before, you can bend the electrodes around or implant the wires first and then run the electrodes into the implant. However, the second option is only available with a reusable transmitter.
All right, Andrew, I have one last follow-up question for you. What is the recommended technique to strip the platinum iridium wires?
So the best technique for stripping platinum iridium wires is probably thermal stripping. It lessens the chances of wire being damaged and is usually easy for users to execute. So there's two ways to strip EEG wires. One is just simply use a cigarette lighter or a flame, where the wire that you'd like to strip is briefly touched to the flame, and the insulation cleanly burns off the electrode. However, don't try using this technique on the ECG or EMG wires, as it just doesn't work. So the other technique is to use a professional tool, which is made by Hacko. It's a thermal stripper. So what you do, it's basically forceps. They get heated up, which then lets you strip the insulation from the wire at the very precise location. Uh, the tool will work for both platinum iridium electrodes and stainless steel electrodes that are used for EMG. So use as your own caution.
Okay. Well, thank you so much. I know that was a lot of questions all at once, and we wanted to just like get those taken care of before we head over to Fraser. So Fraser, I'm going to go ahead and make you presenter. Thanks again, Andrew.
Thank you.
Okay, thanks, Brenda. So this section I'm going to talk about data analysis, and in particular looking at the tools that are available within the Acknowledge software. So if you're using Acknowledge or our data acquisition systems, Acknowledge comes with a copy of with your device. It's basic software for displaying and analyzing data. It's a whole slew of automated analysis routines and filters, both online and offline. It allows you to record video so that you get the behavioral information as well as the physiological data. A lot of the things that I'm going to show you today, they can also be fully automated using workflow. Workflow is it's a licensed feature that allows you to create automated routines using a drag and drop user interface. So there's no programming required. You basically drag over the tools that you want to use. You'll open up the dialog box in a similar way to the normal operational mode within Acknowledge, set it all up, and then when you run the workflow, it can be run across a single channel, multiple channels, or across multiple files. So it's a really convenient way of being able to automate a lot of those routine procedures that you're likely to incorporate into your procedures. Okay, let's take a look at Acknowledge then. This is the Acknowledge software. Here we're showing a single channel of EPOCH EEG data. And if you look, you can see that there's some spike activity in there. One of the nice things about Acknowledge is you've got total flexibility. You can manually control the vertical and horizontal axes so that you can zoom in and get a better look. You can go back using the toolbar to where we were before. You've got a magnifying glass that just allows you to draw over an area, zoom in, get exactly the view that you want. Using the software, if I get this set up correctly, we can just very quickly eyeball the data to see what was going on at any moment in the recording, and we can get back to where we were. Other nice tools within the software, I've selected all the data and I'm just going to duplicate this channel. So I used my right mouse button. I've got two identical channels, and maybe I want to run a filter across here. So under digital filters, FIR, I'll use a bandpass filter to tease out the alpha wave activity. So between 8 and 13 hertz. Filtering the entire waveform. And now we've got this data, this waveform here, which is showing the alpha wave activity within the data. If I want to remove this channel, I can put my finger on the Alt key on the keyboard and just hide it. So I haven't deleted it, I've actually just hidden it. Under the analysis menu, there's a sub-menu down here for EEG, and there are tools such as deriving the EEG frequency bands, so we can pick the one channel, hit OK. Now the software will go through and calculate alpha, beta, delta, theta, gamma waves and display them for us. So very quickly we've got those channels. So kind of automated way of being able to get at that information. Also, there's a really nice tool in here for looking at the seizures. So I'm going to set this up. This is going to create a new waveform, a second waveform, which is going to be a difference channel. And I'm going to use a threshold of 40. I'm going to use a 60-second window, and I'll explain what this is. So what's going to happen, the software will identify all the seizures that exceed 40 millivolts in the difference channel, and then it will run through and look at each 60-second window and count the number of spikes. If the spike count exceeds 30, then it will score that area as a seizure. And what I'm going to do is create a focus area for each area, each 1 minute period that contains a seizure. I'm going to output events, and I'm going to create three new channels, plus output the results to Excel. So hit OK. Okay, perfect. So let's move this over. The top channel is the raw EEG, and you can see these check marks, and I'll zoom in so you get a better view of an area like that. These check marks are based on the difference channel exceeding the threshold that I set. And if you remember, I set a threshold of 40 millivolts. So anytime this pink channel down here exceeded 40 millivolts, it's identifying that spike in the raw EEG. If I compress back down to where we were, this third channel, and actually if I take this, is red, this red channel. This represents a spike count per 60-second period. So you can see this is being updated every 60 seconds with a new count. And then the bottom channel, which we'll be able to see a little bit easier, I'm going to turn the focus areas off temporarily. Each time this channel goes high and remains high, that's indicative of a seizure episode. So in this 1 minute period, we crossed the 30 spike count that I selected. And the reason why we couldn't see these before is because I selected to use focus areas. So the focus areas, completely hide those. So focus areas are labeled for us, seizure 1, 2, 3, 4, 5, so representing the focus areas. The focus areas are helpful because we can use those for further analysis. Okay, if I come back over to the Excel output, we get a summary of how I had everything set up. So we had a threshold of 40, we had frequency of 60 seconds or window width of 60 seconds and our threshold of 30 spikes. These are the episodes that contained a seizure, 36, 31, 37, 41, 140. First column here represents the time windows starting at 0, 60, 120, 180, so on and so forth. Then the spike count that occurred within that 60-second window, and then seizure detected. This is just binary. There, 0 represents no seizure, 1 there was a seizure. So here, for example, at this time window 1920, 36 spikes were counted. That exceeded our threshold of 30. So we see a 1 indicating a seizure. So pretty nice output there. That gives you a quick overview of the seizure detection. Something else that you can do within the system when you're collecting data is come in and capture the video. So I've got a separate camera down here. This can be facing the cage or mounted on top of the cage looking down at the animal. The moment we start to record data, the video is time synchronized with the physiological data. So when you place the cursor in the physiological recording, it will advance the video to the corresponding frame in the video. And if you advance the video to an area that you're interested in, it will place the cursor at the corresponding point in the physiological record. The tools available also include FFT, power spectral density. So if you want to run an FFT, different ways of doing it, you can select an area, come into the FFT dialog. You've got logarithmic or linear outputs, hit OK, and then the system will provide you with the FFT for that area. You can do the same if we do select all. Actually, I was looking at the wrong channel. If we do that for power spectral density, hit OK, and now we've got the power spectral density for that entire 1 hour recording of raw data. So it's easy to get access. Video is easy, different types of analysis, both time and frequency domain, dependent upon how you want to look at the data. Another tool which can be super handy is there's a spectrum analyzer palette. This will perform the spectral analysis over the selected area, or it can be performed in real time. So if this was a real-time recording, we would see the analyzer display the frequency components of that recording in real time. So super convenient. Also, we have the event palette. So each one of those check marks is labeled as response. We get the time, you can see all of those, and you can jump to any event within the recording. All right, so let's come back over to my PowerPoint. Finally, the acknowledged files can be opened up within SleepSign. SleepSign is a fully automated program for scoring and staging sleep in rodents. Beauty of it is it has a really nice graphical user interface. It's fully automated and it's extremely fast. So a 24-hour file can be analyzed in approximately 5 minutes. You can go through there and view and make adjustments to your settings as well by, sort of doing a manual scoring. But once you've got it all set up, the system is nicely automated. So finally, I want to give you a little bit more information about some of the development work that we're working on. BioPac and the University of Utah are collaborating on an SBIR Phase 2 grant. This was awarded to us by the NIH and National Institute of Mental Health, which very fortunate to have that grant. And we're using it to develop a chamber specifically for the PUP system. So there are several goals of the grant, but the most fundamental part of the grant is developing this chamber. The reason for doing that is immature rodents are central to modeling human neurological disease, yet inconsistency in environmental and physiological monitoring hampers reproducibility. Having a sensor small enough to record from pups is only part of the problem, which is why we're working on this PUP chamber. We're aiming to provide researchers with an out-of-the-box solution for rodent pup recording of EEG data that offers tight control and should afford better reproducibility across animals, studies, and labs. Challenges in immature rodent research really revolve around the pups not being able to thermoregulate until P12. So poor temperature controls alters lesion size and mortality. Wired EEGs induce stress, and in fact, it's almost impossible to do a tethered system with a pup because the skulls just haven't formed properly. They're too soft. So this chamber will allow us to provide precise and tight control of the environment. We're using precise Peltier-based temperature controller, and that will monitor temperature, control temperature, and also it will regulate humidity as well. You can run multiple chambers to one NP200 system, standardized protocols and temperature recovery profiles. It's got built-in alarms and remote monitoring, so everything is controlled on the front panel here. You've got analog outputs for chamber temperature, chamber humidity, and then the physiological data that's coming from the animal. For hypothermia, which is routinely required for certain clinical protocols, brain injury protocols, the Peltier device will not only provide heat, but it will cool as well, very precisely controlled and then very rapid in adjusting those temperatures. So the idea is trying to support translational neuroscience and drug discovery with the introduction of this PUP chamber. We hope to have it available in 2026, next year. Okay, one last thing from me before I hand back over to Brenda. Andrew has provided an incredible walkthrough of the surgery that's required for these. BioPack does offer surgical training. So if you're interested in setting up training in your lab, let us know. Some QR codes there. Or if you're interested in just general training on BioPack, you can let us know and we'll definitely help you out.
Well, thank you, Fraser and Andrew. Really appreciate the time that you took to put this presentation together. It was very interesting, and I know the audience appreciated your work on this. I want to let everyone know that we host webinars almost every month of the year. We have over 100 webinars on our website, and you're welcome to watch those at your convenience. Also, Fraser mentioned that we can host a surgery training, but we also can just bring any type of equipment to your regional location, and you can host a workshop in your area and invite your regional colleagues. It's a great way to learn about the products, get your hands on the products. It's usually a half a day or a full day or multi-day if you're interested in that. We've hosted we've hosted workshops all over the world. And we're happy to talk to you about hosting one in your area. Just reach out to our representative and you can start a conversation. And finally, I just want to conclude by saying today you may visit biopack.com at any time for additional references, including screencasts, application notes with step-by-step instructions, and of course, information on upcoming events. We will be at Neuroscience, so please come join us and say hello. Come by our booth and say hello if you're at Neuroscience this year or any year. Thank you all for participating. Thank you again to our presenters, Andrew and Fraser. Have a great rest of your day, everyone. Goodbye.